By the end of this unit, you should understand the structure of bone, its major functions in the body, how bone is formed in the fetus, and how it is remodeled throughout life. The name of this organ systems comes from the word skeletos, which means dried up. It is so named because this is how it appears after death when all the living tissue has decomposed. Bone or osseous tissue in living beings is incredibly dynamic and is constantly being remodeled. The functions of the skeletal system include working in conjunction with the muscular system to create movement, support for the body, mineral storage, blood cell formation, and protection of vital organs. Bones and their surface markings, such as bumps, ridges, grooves, tubercles, etc., serve as anatomical, clinical, and surgical landmarks. Many important bone surface markings are sites of muscle and ligament attachment. Examples include the greater and lesser trochanters, which are large, blunt, and irregular processes found only on the femur. These serve as attachment sites of large, powerful muscles that move the thigh. Tuberosities are rounded, rough projections, such as the deltoid tuberosity seen on the humerus in the diagram above. Tubercles are smaller rounded projections, such as the adductor tubercle shown on the distal end of the femur. A crest is a prominent narrow ridge of bone as exemplified by the iliac crest, which is the structure we rest our hands on when we put them on our hips. A line is a small, narrow ridge of bone, and a spine is a sharp, slender, often pointed projection. A second category of bone surface markings include the projections that help form joints or articulations. The head of a bone is an expansion carried on a narrow neck. A facet is a smooth, nearly flat articular surface. A condyle is a rounded protuberance at the end of a bone articular projection. A ramus is an arm-like bar of bone that runs in a different direction from the main body of the bone. Examples are found on the mandible and the pubic bones. The third category of bony landmarks are depressions and openings that allow blood vessels and nerves to pass. A meatus is a canal-like passageway, such as the external auditory meatus. A sinus is a cavity within a bone filled with air and lined with mucous membrane. A fissure is a slit-like opening, and a foramen is a round or oval opening through a bone. A shallow depression in which a structure like an organ or muscle may sit is called a fossa. At birth, the skeleton is made up of about 270 bones. A number of these fuse during the first years of life, so that the adult skeleton consists of 206 bones. These bones are comprised of two basic types of osseous or bone tissue, compact and spongy. Compact bone is dense and looks smooth. It is found on the outer part of all bones. Spongy bone is composed of small, needle-like pieces of bone referred to as spicules or thin, flat plates of bone called lamellae. Spongy bone also has space between the spicules. In addition to classifying bones based on their microscopic anatomy, bones are classified based on what they look like. There are four principal types of bones based on their shape: long bones, short bones, flat bones, and irregular bones. Long bones are longer than they are wide. They can range greatly in length. For example, the femur is a long bone and has the distinction of being the largest bone in the body. But the phalanges of the fingers and toes are also long bones. Short bones are roughly cube-shaped, meaning that their width and length are somewhat equal. They are made up mainly of spongy bone, except on the surface. Examples include the 8 carpal bones that make up the wrist. Flat bones are thin and consist of 2 parallel plates of compact bone with spongy bone sandwiched in the middle. The main functions of flat bones include protection. They also provide a large surface for attachment of muscles. Examples include the cranial bones, sternum, and scapula. Irregular bones have complex shapes and can't be easily classified. They possess varying amounts of compact and spongy bone. Examples include the vertebrae and facial bones. Long bones have three major parts: the diaphysis, epiphysis, and metaphysis. The diaphysis, or shaft, makes up most of the bone's length. It is covered and protected by periosteum dense, irregular CT. In addition, the diaphysis is made up mostly of compact bones surrounding a central marrow cavity. The contents of the cavity is referred to as yellow marrow because it is filled with adipose tissue which, in the fresh state, appears yellowish. The epiphyses are found making up both ends of the long bone. The part of the epiphysis that is found in a joint is covered by a thin layer of hyaline cartilage called the articular cartilage. The spongy bone in the epiphysis contains red marrow and is the site of blood cell formation or hematopoiesis in adults. The metaphysis connects the epiphysis to the diaphysis. This is an anterior view of the humerus. The proximal half of the long bone was cut away longitudinally. Notice that there is a thin line of bony tissue in the epiphysis. This structure is called the epiphyseal line. It is the remnant of the epiphyseal plate, a flat piece of hyaline cartilage seen in young, growing bones. In addition, a small pie-shaped piece was removed from the proximal epiphysis and is magnified in Figure B to show the structure of compact and spongy bone. The picture in the lower right is through the diaphysis. It shows mainly compact bone, yellow marrow, and the periosteum which brings blood vessels to the bone. Short and flat bones look a lot different from a typical long bone. Both types of bones consist of spongy bone sandwiched in between two thin layers of compact bone. In life, periosteum covers the compact bone. The internal spicules of spongy bone or diploe are lined with connective tissue called the endosteum. The term diploe is used to describe the spongy bone found in flat, short, and irregular bones. Some bones are not classified by shape, but instead are classified based on their location. For example, sutural or wormian bones are found within the sutures of some cranial bones. There is a lot of individual variation in these bones. Some people don't have any and others can have 1, 2, 3, or more. There is no clinical significance to the number or presence or absence of sutural bones. Instead, it is related to how these bones develop. Sesamoid bones are sesame seed-shaped bones that are found in a few tendons that pass over a joint. They are found in the hand, especially metacarpophalangeal joint of the thumb, the foot at the first metatarsophalangeal joint of the great toe, and the knee. They protect the tendon by holding it farther away from the joint. This also modifies the mechanical effect of the muscle and reduces friction. Sesamoid bones can alter the direction of a muscle pull across the joint. Like all connective tissues, osseous tissue consists of cells, fibers, and ground substance. Together, the ground substance and fibers are called the matrix. Cells account for only 2% of the mass of bone. Collagen fibers give bone its strength and account for about 33% of bone mass. The ground substance is made up of chondroitin sulfate and has been hardened by the infiltration of calcium salts, collectively referred to as hydroxyapatite. The mineralized ground substance accounts for 2/3 or 66% of the bone mass. It makes bone hard so it can carry out its function of supporting the body. But mineralization also makes bone inflexible and brittle. There are 4 types of living cells found in bone. Osteocytes bone cell are mature bone cells. They cannot divide or secrete bone matrix. The osteocytes sit in spaces called lacunae and form connections with other osteocytes via small channels in the bone called canaliculi. The osteocytes in an osteon are connected to each other through gap junctions which permit the passage of materials between the bloodstream and the cells. Diffusion doesn't work in a mineralized matrix. The function of osteocytes is to maintain the integrity of the protein and minerals of bone. The second type of cell is called an osteoblast. These are immature bone cells that cannot divide, but they secrete unossified bone matrix called osteoid. Once the osteoblasts are surrounded by ossified bone matrix, they become mature bone cells or osteocytes. Osteoprogenitors are bone stem cells that give rise to osteoblasts when new bone is needed. They have the ability to divide and are important in repairing fractures. Osteoblasts are found in the inner layers of periosteum and endosteum. Osteoclasts bone break are large cells containing over 50 nuclei. They were formed by the fusion of blood monocytes and tissue macrophages. Their job is to dissolve bone matrix. They play an important role in bone remodeling and turnover. The anatomical and functional unit of compact bone is the osteon or Haversian system. Each osteon is comprised of concentric rings or lamellae of bone surrounding a central canal. Central canals, containing the blood supply and nerves, run parallel to the surface of the bone. Perforating canals run perpendicular to the surface, penetrate the compact bone, and supply blood to deeper osteons. Bone is highly vascularized because its mineralized matrix prevents diffusion. This slide shows a diagram of the main structural features of compact bone. In the lower right-hand corner, there is a micrograph of a piece of compact bone that was ground very thin and placed on a slide. The slide in your box is similar to this one. As previously stated, osseous tissue must be highly vascularized because the calcified bone matrix makes diffusion impossible. This is particularly true for compact bone. The diagram of the long bone above shows how blood vessels enter and leave a bone. The periosteal vessels gain entrance to superficial osteons. The nutrient artery and vein bring blood into the inner regions and the marrow cavity. From these sources, small vessels as well as nerves enter the central canals of the osteons of compact bone. Arteries bring oxygenated nutrient-rich blood to the osteons, and these materials are conveyed via the gap junctions that connect the osteocytes of an osteon. Carbon dioxide and other metabolic waste generated by cellular metabolism flow in the opposite direction in the osteon, that is, from the outer lamellae toward the vein in the central canal. All osseous tissue is dynamic and ever-changing in response to many stimuli. Compact bone, in particular, thickens along lines of stress. Weight-bearing exercise, pulling of muscle on bone, results in thickening of the bone in the regions of tendon attachment. In addition, long bones like the femur are subjected to bending stress from supporting the weight of the body. Body weight transmitted to the head of the femur threatens to bend the bone along the arc shown by the dashed line in the picture above. The bending compresses the bone on one side indicated by the two red converging arrows, and stretches it on the other side shown as blue diverging arrows. Because these two forces cancel each other internally, much less bone material is needed internally than superficially. Thus, the shaft of the femur can withstand 10 to 15 times the body's weight. Osteons run parallel to the long axis of the shaft. Bones are broken from a lateral impact to the shaft perpendicular to the os. The terms spongy bone, trabecular, and cancellous bone all refer to the same type of osseous tissue. The lamellae of spongy bone are not arranged in osteons. Instead, they are arranged in bony spicules called trabeculae. These branch forming an open network. There is no need for blood vessels or capillaries to run through trabeculae because of the numerous blood vessels running in between them. Osteocytes are still connected to one another via gap junctions, and canaliculi are still present. The presence of spongy bone reduces weight of the skeleton. Red marrow is found between trabeculae in the epiphysis of long bones, and is the site of hematopoiesis blood cell formation. Yellow marrow adipose tissue is found between the trabeculae in the marrow cavity of the diaphysis. The process of bone formation is called ossification. Ossification occurs in two ways. endochondral in cartilage and intramembranous within membranes. The skeleton begins to form 6 weeks following fertilization. The skeleton of embryos is made largely of hyaline cartilage except for flat bones which develop in fibrous membranes. In endochondral ossification, a cartilage model is made first, but by the time of birth, it has been largely replaced by bone. In the picture of the chick embryo above, the parts of the skeleton that stain blue are still cartilage, whereas the parts that have already ossified stain red. The diagram above depicts the steps occurring during endochondral ossification. In the first step, a bony collar forms around the hyaline cartilage model of a bone. Shortly afterward, the cartilage matrix in the center of the shaft begins to die and deteriorate. Next, blood vessels from the periosteum invade the cavities created within the bone. As ossification continues, the medullary cavity enlarges and secondary ossification centers are formed in the epiphysis. At birth, only 2 pieces of the original hyaline cartilage model remains. The articular cartilage, which is permanent and persists throughout our lifespan, and a small band of cartilage forming the epiphyseal plates at either end of the long bone. It is the epiphyseal cartilage that facilitates growth in length of bones. This is how a 19-long infant can grow into a 6'5" man. Bones grow in length because certain hormones. Growth hormone, secreted by the pituitary gland, keeps the chondrocytes healthy, happy, and dividing on the epiphyseal side of the growth cartilage, whereas just a millimeter away on the diaphyseal side, the cartilage cells are swelling up and dying. The calcified cartilage matrix is ultimately replaced by bone. This slide illustrates how bones grow in length via the epiphyseal growth cartilage. Cartilage cells are dividing on the epiphyseal side and dying and being replaced by bone on the diaphyseal surface. Above is an X-ray of a child's hand on the left and an X-ray of an adult's hand on the right. Notice the spaces indicated by the arrows in the bones of the child's hand. Cartilage does not show up on X-rays, so the sites where the epiphyseal cartilages are located appear as an empty space. The X-ray of the adult hand shows where the epiphyseal cartilages were prior to adolescence. They now are solid bone and appear as white lines on the X-ray. If growth via epiphyseal cartilages was the only way bones grew, then we would have long but very skinny bones that could not support our weight. This doesn't happen because bones also grow in width, which matches their growth in length. Growth in bone width occurs by appositional bone formation, or growth on the bone surface. To have this happen so that the correct proportions of the bone are maintained as it grows in both length and width requires the cooperative activities of osteoblasts, which synthesize bone matrix, and osteoclasts, which dissolve it. Osteoblasts in the periosteum add bone tissue to the external face of the diaphysis, as osteoclasts in the endosteum remove bone from the inner face of the diaphysis wall. Intramembranous ossification is how most flat bones are formed. In this type of ossification, there is no model of cartilage. Instead, ossification centers appear in fibrous connective tissue membranes. Recall that mesenchymal cells are embryonic cells that can become other types of CT cells even in the adult. In intramembranous ossification, mesenchymal cells differentiate into osteoblasts shown in yellow above and begin secreting osteoid. The osteoid mineralizes in a few days and the osteoblasts become entrapped in it. At this point, they become mature bone cells, or osteocytes. New osteoblasts begin adding to the surface of the ossified bony spicule, and the spicule gets bigger and bigger, entrapping more cells. Next, the membrane condenses, forming the periosteum. The accumulating osteoid is laid down between embryonic blood vessels, and compact bone forms on the surface of the trabeculae. This is a histological section through the skull of a fetal rat. The surfaces of the smooth red bone trabeculae are coated with osteoblasts that appear as small dark dots under this low magnification. They continue to lay down bone. No compact bone has been formed at this stage. The growth of the skull bones is perfectly adjusted to the growth of the brain. This is because the mechanical forces caused by the pressure generated by the expanding brain influence the activity of the osteoblasts. As brain growth slows, so does ossification. Babies are born with soft spots called fontanelles. These are spaces between the bones of the skull containing fibrous CT membranes. The fontanelles allow for the brain to keep growing after birth and are supposed to make parturition, vaginal birth, easier. Notice the raised, cone-shaped areas on the skull shown by the green arrows. These are the original sites of intramembranous ossification. They are primary ossification centers. This slide shows the skull of a 12-week-old fetus. The reddish web-like strands emanating out from a central core is bone growing via intramembranous ossification.